A very good evening to everyone present here. I welcome all the participants on today's session. Let's welcome today's speaker with great honor, none other than Dr. Kumar Prafulla Chandra sir. Let me share some of the achievement of the sir. Sir has done MBBS, MD in medicine, DFID from CMC Vellore, PGDM Advanced Diabetic Care, UK, Director and Department of Internal Medicine and Diabetes Care, Health City Vistar Hospital, Gomti Nagar, Lucknow, and Director Chandra Diabetes and Obesity Clinic, Vijay Khand, Gomti Nagar, Lucknow. And for today's session, the topic is antimicrobial resistance. And before starting this session, let me tell you overview of the topic. Eye-opening webinar as delve in the critical issue of antimicrobial resistance, that is AMR. Antimicrobial resistance poses a significant threat to the global public health, rendering one's effective medication ineffective and complicating treatment of the infectious disease. To know more about this topic, I would like to invite sir and hand over this session to sir. Over to you, sir. Kindly proceed from here.
Thank you very much, IJCP team, for giving me this opportunity to talk on about antimicrobial resistance. The topic is important because the present week that we are in, that is from 17th November to 24th November, is Antibiotic Awareness Week. And here, during this week, we need to understand what should be the proper use of antimicrobials and how to prevent resistance. So, moving ahead, the first thing that we need to understand is what is antimicrobial resistance, and I will try to talk about this topic in detail. Basically, I would be talking about definitions, what actually antimicrobials are, what is role of antimicrobials, what is the cause of resistance, what is the meaning of resistance. So basic things. I mean, we keep on reading and learning a lot of things, but we tend to forget few basic things about the antimicrobials, and I will try to elaborate upon those aspects here in today's deliberation. So, what does it mean, antimicrobial resistance? So, let me tell you that antimicrobial resistance happen when germs like bacteria and fungi develop ability to defeat the drug designed to kill them. What does this mean? This means that germs are not killed by those antimicrobials which were designed. I mean, those had to kill them, but it continues to grow even in presence of those antimicrobial. And let me tell you, when this antimicrobial resistance develop, it is usually difficult and sometimes impossible to treat an infection caused by resistant microorganism, which may include either bacteria or fungi or both. Antimicrobial resistance does not mean that our body is resistant. Many times, you listen people saying that my body is resistant to antibacterials. So that is not correct. Our body doesn't have any role. Our body doesn't develop resistance to antibiotic or antifungals. Basically, the bacteria and fungi that causes infection to our body are resistant to antibiotic and antifungal treatment. Now, fungi and bacteria do not have resistance to each and every antibiotic or antifungal to become dangerous. So even resistance to one antibiotic may cause a serious problem. Let me give you a couple of examples. Antimicrobial resistance infection that requires use of second or third line treatment can harm patient by causing serious side effects such as organ failure, prolonged care or recovery. And there are certain conditions in which our immunity is compromised. There are certain devices, and under those conditions, many medical advances are dependent on ability to fight infection using antibiotics. Like a person has got joint replacement, and if there is an infection which is resistant to antimicrobial, it will be really difficult. Organ transplant, cancer therapy, treatment of chronic illnesses like diabetes, asthma, rheumatoid arthritis. In these patients, basically any infection may be life threatening. And in those conditions, if there is resistance, it's really a tough job. And in some cases, where their disinfection develop, then we are left with no treatment options. So this is what antibacterial resistance means. Now, how does antimicrobial resistance happen? So antimicrobial resistance happen when germs like bacteria and fungi develop ability to defeat the drug. So resistant infections are difficult and sometimes impossible to treat. Let me tell you that antimicrobial resistance occur naturally. However, increase in the antimicrobial resistance are driven by combination of germ exposed to antibiotics and antifungals and spread of those germs and their resistant mechanism. So what happens that there is some resistance already there in some bacteria or fungi. When these bacteria or fungi grow out of proportion and infection is caused mainly because of those bacteria or fungi which are inherently having resistance, then we call that the infection has become resistant to antimicrobial treatment. Now, before I go ahead, few basic things. Why antimicrobial? What are germs? So germs we all know. These are bacteria, fungi or viruses which infect us. These are small organisms. And I will tell you that there are many small germs which are harmless to us, even helpful. We all say normal commensals. In our gut, they are normal lactobacillus bacteria. And some cause infection. And those germs which cause infection are called pathogens. And when we use antibiotic, we try to kill those pathogens. But what happens then when you take an antimicrobial, it does not differentiate between pathogens and inherent or helpful bacteria. So it kills all. So antimicrobial terms are used to describe drugs that treat many types of such infections by killing or slowing the growth of pathogens causing infection. Bacteria cause infections such as sore throat, foodborne illnesses, other serious illnesses. Antibiotics are there to treat bacteria. Fungi cause infections like athlete foot, yeast infection, and other serious infections, candidiasis. And there are many fungal infections nowadays, we all know, and antifungal treat fungal infections. Now, how this antibiotic and antifungal use affects the resistance? We all say that we are using antibiotics, antifungals inadvertently, and that is leading to resistance. Let us understand why this resistance develops. So, antimicrobial resistance is accelerated when the presence of antibiotic and antifungal pressure bacteria and fungi to adapt. So, in a milieu of antibiotic or antifungal, those bacteria or fungus which are there, they start adapting. They adapt to grow even in presence of those antimicrobials. Antibiotics and antifungal kill some agent that causes infection, but they also kill helpful germs that protect our body from the infection. So this antimicrobial does not differentiate in between pathogen and helpful germs, and it causes both, kills both. Now, what happens that antimicrobial resistant germ survive and multiply? So, out of say there are, I'm just giving an example. There are 100 pathogens. There might be 95 pathogens which are killed by the bacteria. Only 5 remains. Now, what happens that when you have taken antibiotic, even helpful germs or helpful bacteria which was protecting your body is also killed. So those 5 bacteria which was not killed by the antibiotic has survived. Now, the natural immunity is not there because the antibiotic has killed helpful bacteria also. So, those five resistant bacteria which has survived starts multiplying, and that is how those surviving germs which has got resistant traits in their DNA spreads, and ultimately infection with resistant microbes does develop. And this is how mycobacteria or antimicrobial resistance develop in the patient. Now, how does this germ spread and what are the different resistance mechanisms? So, what happens to survive? Germ can develop defense strategies against antibiotic, antifungal, that is called resistance mechanism, and there are different mechanisms for it. DNA tells the germ how to make a specific protein which determines germ's resistance mechanism. Bacteria and fungi can carry genes from many types of resistance. When already hard to treat germ has a right combination of resistance mechanism, it can make all antibiotics or antifungals ineffective, resulting in untreatable infection. And these are the infection what we call superbugs. I mean those bacteria which cannot be treated by any antibiotic. And this happens because there is, inherent some resistance and some right combination of different resistance mechanism occurs in a single bacteria, and those combinations lead to such a change in bacteria that it cannot be killed by any antibiotic, and then we call it superbug. So this is how resistance mechanism develop in microbes. Alarmingly, antimicrobial resistant germ can share its resistance mechanism with other germs that has not been exposed to antibiotic and antifungal. This is another important point. So if one bacteria is having resistance, it can transfer the resistance to other bacteria which was earlier sensitive. And we will try to understand what are the different mechanisms by which this antimicrobial resistance is transferred from a resistant strain to a sensitive strain and how this resistance spreads. Now, moving ahead, how the resistance is spread. I just told you, this is just recap of what I have told. Germ, bacteria and fungi are everywhere. Some help us, some make people, crops and animals sick. Antibiotic kill germ that cause infection, but antibiotic resistance germ find the way to survive. Antibiotic resistant germ can multiply. Some resistant germ can also give their resistance directly to the germs. Antibiotics also kill helpful germs that protects us. Without the helpful germ, resistant germ has even a bigger advantage to spread. And once antibiotic resistance emerges, it can spread into new setting between the different countries. This is how resistance spread. Now let us understand what are the resistance mechanism or defense strategies that a bacteria has against the antibiotic. So first is restrict the access to the antibiotic. So when an antibiotic is there, it will have a limited entry into the germ, bacteria or fungi. So that how it is, germ restricts access by changing the entryway. The way antibiotic would enter into the bacteria, that entryway is changed or limiting the number of entryways. There are a couple of examples. Gram-negative bacteria has an outer layer membrane that protects them from their environment. These bacteria can use this mechanism to selectively keep antibiotic drugs from entering. The second mechanism is get rid of antibiotic or antifungal. So germs get rid of antibiotic using pumps in their cell wall to remove antibiotic drug that enter the cell. So antibiotic has entered, there is a pump, it will pump it out. So there is no antibiotic inside the cell and bacteria will survive. Some examples include Pseudomonas aeruginosa bacteria that can produce pump to get rid of several different important antibiotic drugs, which include fluoroquinolones, beta-lactams, chlorphenicol, and trimethoprim. There are some candida species produce pumps that get rid of azoles such as fluconazole, and that's why you can see that many times we are having fluconazole resistant candida infection. Now, the third mechanism, change or destroy the antibiotic. So there can be some enzymes or some proteins which can be produced by these germs, bacteria and fungi, which will destroy the antibiotic and break down the drug. And that happens in Klebsiella pneumonia bacteria, which produces enzyme called carbapinamase or penicillinase, which breaks down the carbapinam drug and most other beta-lactam drugs. So this is how resistance develop. Then comes another mechanism, change the target of antibiotic or antifungal. Many antibiotic drugs are designed to single out and destroy the specific part or target of the bacterium. Germs change the antibiotic target so the drug can no longer fit and do its job. Once again, example, E. coli bacteria with MCR1 gene can add a compound to the outside of the cell wall so that the drug colistin cannot latch onto it. So antibiotic has to enter bacteria through a port. Now, if you change the port, the entry will be restricted, and that is what it happens in E. coli with mutation in MCR gene. Aspergillus fumigatus fungus changes CYP1 gene so that triazole cannot bind to the protein. Another mechanism, bypass the effect of antibiotic. Germ develop new cell processes that avoid using antibiotic target. Example is Staphylococcus aureus bacteria can bypass drug effect from trimethoprim. Now, these are the different mechanisms. Once again, I have already discussed. This is a repetition. How bacteria or fungi fight back the antibiotic. Antibiotic fight germs, bacteria and fungi, but germs fight back and find new ways to survive. Their defense strategies are called resistance mechanism. Only germs, not people, become resistant to antibiotic. So germ develop new cell processes that avoid using antibiotic targets. Germ change or destroy the antibiotic with enzymes. Germ restrict access of antibiotic to the entry pathway. Germs get rid of the antibiotic using pumps, and germ change the antibiotic target so the drug can no longer fit and do its job. Could you try again? Now, how antibiotic resistance moves directly from germ to germ? How a resistance which is in one bacteria goes into the another bacteria. So there are different mechanisms. I'm going to just elucidate that. Any antibiotic use can lead to antibiotic resistance. Antibiotic kills germ like bacteria, fungi, but resistant survivors remain. Resistant traits can be inherited generation to generation. So once it multiplies, the next generation will have resistance. They can also pass directly from germ to germ by way of mobile genetic element. And what are these mobile genetic element? First is plasmid. Plasmid is circle of DNA that can move between the cells. Then comes transposons. These are small pieces of DNA that can go into and change the overall DNA of the cell. These can move from chromosome, which carry all the genes essential for germ survival, to plasmid and back. Then mobile genetic element include phages. So bacteria phage are the viruses. Viruses that attack the germ can carry DNA from germ to germ. Now, how these mobile genetic element work? There is another mechanism called transduction. In transduction, resistant gene can be transferred from one germ to another germ via phages. Conjugation, resistant germ can be transferred between germs when they connect. And transformation, resistant germ released from nearby live and dead germ can be picked up directly by another germ. So this is how resistance transfers from a resistant bacteria or fungi to the non-resistant bacteria, and that is how it spreads. Germ has developed over the time. This is the chart which says when an antibiotic was developed and how the germs developed resistance to them. So you can see it is more than 90 years when penicillin was discovered, 1941, And resistance started developing 1942, 1967, 1976. And you can see a series of drugs written here: vancomycin, amphotericin B, methicillin, extended spectrum cephalosporins, azithromycin, imipenem, ciprofloxacin, fluconazole, capsofungin, daptomycin, ceftazidime, avibactam. You can see as the drugs are developing, antibiotic resistance are also developing simultaneously and is becoming evident. So while antibiotic development has slowed down, antibiotic resistance has not slowed down. And a type of resistance has developed even against most powerful drug. And this is what the actual problem is. Now, we need to know five most important thing about antibiotic resistance which we have discussed till now. The first is antibiotic resistance occur when germ defeat the drug designed to kill them. Antibiotic resistance can affect people at any stage of the life. So, infection caused by resistant germ are difficult, sometimes impossible to treat. In many cases, these infections require extended hospital stay, additional follow up, doctor's visit, and use of treatment that may be costly, potentially toxic to the patient. Healthy habits can protect you from infections and help stop germs from spreading. So, now comes another important aspect: how can you stop antimicrobial resistance and how can you stop germ to spread? And that is healthy habits. Get recommended vaccines, keep hands, wounds clean, take good care of chronic conditions like diabetes. That is where you will stop the spread of the resistance. Antibiotics save human animal lives. Anytime antibiotics are used, they can lead to side effects and resistance. Antibiotics do not work on viruses such as cold and flu. Talk to your healthcare provider or when a veterinarian when you use antibiotic in common cold. Antibiotic resistance has been found in all regions of the world. Now we had COVID pandemic. It had a huge impact on antimicrobial resistance. We'll try to understand how COVID-19 has had its impact on antimicrobial resistance. So, antibiotic use throughout the pandemic varied across the healthcare setting. Antibiotics were commonly prescribed to patients with COVID 19, even though antibiotics are not effective against virus like one that causes COVID 19. So, there were many formulas, azithromycin is working, doxycycline is working, clarithromycin is working, hydroxychloroquine is working. So, people kept on using antibiotic in COVID 19. However, the fact is that none of the antibiotics are known to kill COVID-19 virus. Almost 80% of the patients hospitalized with COVID-19 received an antibiotic. Antibiotics and antifungal can save life, but anytime they are used for people animal, they can contribute to resistance. And in hospital, approximately half of the hospitalized patients received ceftriaxone, which were commonly prescribed apart from azithromycin. So actually there were issues. There were at times people were not able to differentiate in between COVID-19 pneumonia and community acquired pneumonia, and these antibiotics have been used. So when you use antibiotic when it is not needed, certainly those bacteria which are resistant to these antibiotics would develop, and that is what it happened during COVID-19. In outpatient setting, antibiotic use significantly dropped later part of the infection, but azithromycin is what it was used very high, and use of azithromycin was about 50% more than what it was before COVID 19. Now, another aspect that we need to understand: our sensitivity to vaccines, therapeutic, and diagnostics improved. This is the another part of the story of COVID-19. So COVID-19 pandemic highlighted the need to stop the spread of germ before they can cause infection. And that's why you will remember that during COVID 19, other infections were reduced: tuberculosis, pneumonia, other infections. Since a lot of hygiene practice came into day to day activities, other infections stopped. So, COVID 19 pandemic really highlighted how to stop the spread of infection. treatment after infection occurs is not only the solution and should not be the only option. These include alternative to new antibiotic and antifungal drug, new vaccines to combat infection that can develop antimicrobial resistance, novel decolonizing agent to stop spread of antimicrobial resistance. So the another concept that came in, that it is not the antibiotic only which can stop the disease, stop the spread, use vaccines, use other methods by which the spread of the antimicrobial is stopped. Now we move to another aspect of the antimicrobial resistance, and that is called microbial ecology. So we there is importance of understanding microbial ecology when you talk about antimicrobial resistance. So, once you wish to stop antimicrobial resistance, you should know how germ behaves, how they change, how human behaviors drive the emergence and spread of pathogens, helps public health better anticipate, prevent and treat infection, and helps slow the spread of antimicrobial resistance. So when you know all these medical ecology, it will help in slowing down the antimicrobial resistance and spread of the infection. Then, experts working in microbial ecobiology can help public health by studying the relationship between germs, including the factors, ecological pressure that allows some germs to survive and multiply in competitive environment, whereas other germs within the same species do not survive. So they just learn how these things do affect. Then microbial ecology tells us about infection, antibiotic, and antifungal use. So, a healthy microbiome is important for preventing infection and disease. This is very important. There are some healthy microbes within our skin, within our body, within our gut, which helps to prevent the infection. So, that is equally important. An unbalanced microbiome can put people and animal at risk of infection. Pathogens, including resistant pathogen like Clostridium difficile, can take over and multiply in unbalanced microbes. So we all know Clostridium difficile is present in our body, and when you inadvertently use antibiotic that kill other helpful bacteria, this Clostridium difficile multiply exponentially and causes infection. So we need to learn how this bacteria, how we are living in the ocean of bacteria, and how we should, communicate with bacteria so that these bacteria who are helpful do not become harmful, and that usually occurs because of inadvertent use of antibiotics in an inappropriate way. So body is less able to defend against infection when you use these antibiotics inadvertently. Infections caused by resistant germs are difficult. So those Clostridium difficile which survived the antibiotics, now they are resistant, they multiply, and then you get a serious infection with a resistant bacteria. So that is the importance of understanding microbial ecology, so that the spread of resistance and resistant germs can be stopped appropriately. Now, let me give you some examples. When a person takes antibiotic or antifungal, beneficial, health promoting skin and or gut bacteria or fungi are wiped out in addition to pathogen, resulting in an unbalanced microbiome. It can take weeks to months for beneficial bacteria or fungi to return back to the normal strength that they were. In the meantime, people can also spread the harmful pathogen to others, especially if other person have also have disrupted microbiome. Now, another aspect of antimicrobial resistance is where resistance is spread. What are the places where resistance is spread? And if we know about it, we have to intervene so that spread of the resistance can be stopped. So, how germ can spread in healthcare facilities? Without appropriate infection control action, germ can enter the body and cause infection through procedures and medical devices like catheters, spread to people on surface like bed nails, bed rails, or hands of the healthcare worker. move with patient when they are transferred from one healthcare facility to another or into the community when patient goes home. So many times, patient will get a infection from resistant bacteria in the hospital. Patient becomes better and he is shifted back to home. He carries the resistant bacteria along with him to home from where it spreads in the society. That is how resistant germ spreads. We need to know how it spreads so that you can stop it. When not stopped, these resistant healthcare associated germs can spill over into communities, becoming much harder to control. Germs can also spread in other ways, like fecal waste can carry traces of previously consumed antibiotics, antifungals, and antimicrobial resistant germs. The combination of these drugs and resistant germ in hospital wastewater allows these resistant germs to survive in the plumbing system, toilets, and if they contaminate the water resources, this resistance can spread throughout the community. Now, how germ can spread in community? Germs, including resistant germ, can spread between people, animal, food, and through common activities. For example, germ can spread from food to people, between people and animal without appropriate hand hygiene. So, hand hygiene has to be improved to stop the spread. People can also get infected when traveling, then spread these germs when they return. Other examples include gonorrhea or commonly sexually transmitted diseases. Infection associated with drug-resistant gonorrhea and group A streptococcus is most common cause of sore throat, and it has increased further. So this is how germ spreads in community. How germ can spread? Through food supply. Animal, like people, carry germ in their gut, which can include antimicrobial resistant germs. People can get infected from food in different ways, which include from handling or eating meat, seafood, milk, egg that are raw or are undercooked and are contaminated with resistant germs. So if you consume these foods, you will get infection with resistant germ. From handling or eating fruits, vegetable contaminated by resistant germ. From contact with untreated, uncomposted animal waste, either directly when it gets into water environment, from touching and caring of animal without proper hand washing. So many times, animal carry the germ, you touch the animal, you phone the animal, and then you start eating without washing your hand. So you can get infection. Now, what are the actions which are required to fight antimicrobial resistance? So, addressing this threat requires continuous aggressive action to prevent infection in the first place. So, prevent the infection, then improve antibiotic and antifungal use to slow down the development of resistance and stop the spread of resistance when it does develop. Protect yourself and your family. Know your risk. Ask questions and take care. Keep cuts clean, covered until healed. So you have to prevent infection. Take good care of chronic conditions like diabetes and heart disease. Clean your hands. Keeping your hand clean is one of the best way to prevent infection. Avoid getting sick and prevent spreading germs. Get vaccinated. Vaccine are an important step to prevent infection, including resistant infection. Use antibiotics and antifungals appropriately. Be aware of change in your health. Practice healthy habits around animals. Always clean your hand after touching, feeding, or caring for animals, and keep your animals and pet healthy. Healthcare providers, there are certain actions which they should take to combat resistance. Healthcare providers play an important role in preventing infection and stopping germ from spread of germ. Patient might visit your practice with infection, can get infected when receiving healthcare facility, and that is called healthcare associated infections. These infections can be caused by antimicrobial resistant pathogens that is harmful, which may lead to sepsis and death. And then we need to help prevent infection and their state. We need to ask patients if they have recently visited a care in another facility or travel from other countries. Ensure our patients receive a recommended vaccine. Talk to them and their families about preventing infection, keeping scraps and wounds clean, managing chronic conditions, seeking medical care when an infection is not getting better, and understanding when antibiotics and antifungals are needed. alert the receiving facility whenever you are transferring a patient from your facility to another facility and patient is having severe infection. Give a good summary to them that this patient has all these infections and these are the resistance, so that when they reach the another facility they know the antibiotic pattern, and that helps in prevention. Then comes improving antibiotic and antifungal prescribing. This is very much important. Inadvertent use of antibiotic and antifungals should not be done. Follow clinical and treatment guidelines. Implement antibiotic stewardship to ensure appropriate antibiotic use. Consider fungal infection in patients with respiratory infection that do not respond to antibiotic, for example, valley fever, histoplasmosis, blastomycosis. Watch for signs and symptoms of sepsis. Use clinical resources and guidelines so that you use appropriate antibiotic. You get the cure. You don't help in developing resistance. Perform appropriate diagnostic test to guide antibiotic and antifungal therapy. So wherever possible, take the sample for culture sensitivity. Wherever possible, try to remove the source of infection if it can be removed by surgery, including correct drug. And then you need to find out the diagnostic, what is the correct drug which has to be used, what is the correct dose which has to be used for the correct duration, so that you are able to cure the disease, you are able to kill the bacteria, and you don't allow development of resistance. Then comes be alert and take action. Be aware of the infection and resistant pattern in your facility and community. That is the most important aspect in preventing the development of resistance. Ensure you are notified by lab immediately when antimicrobial resistant pathogens are identified in your patient, so that mechanism has to be important. Inform patient and families if they have antimicrobial resistant infection, as well as sexual partner wherever appropriate, so that you are able to stop the spread of the antimicrobial resistant infection. Now, with this, I'm going to the Antibiotic Awareness Week slogan for 2023. It says, improve antibiotic use, improve health equity. So, this antibiotic use should be done appropriately, and health should be received by all in equal way. And be antibiotic aware, be a smart user, so that you give the best care. That is the way you have to stop the spread of antimicrobial resistance. Thank you very much for your patient hearing. Now we can take up question. Farad.
Thank you so much, sir. First, for giving your valuable time and sharing your knowledge with us and making this session informative one, sir. I definitely think this session knowledge is going to help all the participants who have joined this session. Sir, with all your permission, sir, we have received some question. If you allow, can I take this question with all your permission?
Perfect. Can I stop the presentation now?
Yes, sir. You can stop. I have removed the
Okay.
presentation now. Sir, the
Please
first
go on.
question is from Doctor Shreyashi, and she is asking, in what ways can interdisciplinary approach improve patient outcome and reduce resistance?
See, I told about antibiotic stewardship. You have to find out what is the best way of treating. So, best way of treating, identify the pathogen. So, microbiologist, interdepartmental microbiologist will help you in identifying what is the focus of infection, what type of infection is that, which bacteria is there, and which antimicrobial is best to treat that. Number two, if there is a focus of infection which can be removed surgically, say if I've got abscess in muscle, I may keep on using antibiotics for weeks together, but let me tell you, since blood supply to that region is very small, you will not get good response. So here you need to take care of interdepartmental other departments also. Try to remove the focus. Now, if there is infection in bone, you have to give antibiotics together for 4 week, 8 week, 12 week. Now you know that when blood supply is low, there are chances of development of antibiotic resistance. So wherever possible, resect the focus of infection, so that you prevent development of resistance. I told you there are 100 bacteria, 95 out of them will be killed by those bacteria, 5 will not be killed, and those 5 will have survival benefit, and they will start multiplying, and that is how resistance develop. Now, if you have a focus of infection where antibiotic is not reaching appropriately, and if it reaches, it would kill those bacteria which are sensitive, and those which are resistant will stay there because appropriate amount of antibiotic has not reached, and it will grow. This is where the role of interdepartmental communication occurs. Only antibiotic, only injectable antibiotic, only a physician cannot treat all infection. You have to find out what is the cause of infection, which bacteria is causing infection, use appropriate antibiotic, appropriate dose, appropriate duration. Microbiologist friend will help you here. Now, if there is a resectable infection, your surgeon friend, orthopedician, whichever specialty it is, they will come in and help you in treating the infection. That is how a combined effort is going to help you. Then, people from preventive medicine, they will help in stopping the spread of infection in hospital, in community level. So these are really places where a combined effort from different departments are needed so that you can prevent the spread of antimicrobial resistance. Thank you very much for your question.
Thank you so much for answering it really well and explaining it really well. I hope that the doubt is clear, sir. And moving forward, I can see another question from Dr. Manvinder. He's asking, how do advancement in technology, including point of care testing, impact your approach to diagnosis and treating infections?
This is very good question. Let me tell you, empiric therapy that we used to use, broad spectrum antibiotic to be given. My eye is blindfolded. I don't know what I am treating, and I use a shotgun. But yeah, use an antibiotic, a broad spectrum antibiotic, which will kill everything. Now, how it has helped point of care, you are able to diagnose which bacteria is causing. So what happens? Your eyes are open. You know what is the pathogen, what is the cause, and then you use narrow spectrum antibiotic directed against that pathogen. So the other pathogens which were commensals, which were helpful, are not being killed. Other microbes where resistant germ might have developed because of use of broad spectrum antibiotic, you have not used that. You have used specific narrow spectrum antibiotic directed to that particular pathogen, and this is how point of care facilities helped us in diagnosing the disease and treating it appropriately, and thus it has helped in preventing antimicrobial resistance and its spread. Thank you very much for your question.
Thank you so much again for taking up this question very impactfully and very practically, sir. And moving forward, I will be taking last question from today's session from Dr. Manisha, and she is asking, how do you balance the need of effective treatment with the importance of minimizing resistance?
See, that is how you have to formulate. When you know what you are treating, you will do effective treatment. When you don't know what you are treating, you are not going to do effective treatment, you are going to give a broad spectrum treatment. And when you are not doing effective treatment, you are certainly hitting many things. And when you start hitting many things, there are chances that you will lead to development of antimicrobial resistance. So this is how appropriate treatment helps us in decreasing the spread of antimicrobial resistance. Thank you very much for your question.
Thank you so much, sir, for answering all the question and clearing all the dots of the participant. Sir, as I can see, there is no more question from the participant end. With all your permission, shall we conclude this session for today?
Perfect, thank you very much.
Yeah, sir. Thank you so much, and hope to see you again with different topic in future, sir. Till then, take care of yourself. It was a wonderful session, and it was great to have you on our platform, sir.
It was my pleasure, and I really want to thank IJCP team for giving me this opportunity to interact. Thank you very much once again.
Welcome, sir. It's really like it's our pleasure to have you, sir. Like if the correct knowledge is shared on the platform, that is very nice from the healthcare experts, sir. Thank you once again, sir, taking up this opportunity.
Thank you, bye bye.